Janne. Thank you for joining me today to discuss this forthcoming Day of Immunology on Tregs. I'm very excited to have a Treg expert to talk to, such as yourself. So for starters, can you tell me a little bit about yourself, where you work, what you work with?
Yes. So I am a PhD student, currently in my third year of my PhD, and I work in Belgium in Hasselt University. So it's in the east of Belgium. We're a very small university, very cozy. And here I work under Professor Bieke Broux. And I mainly work on regulatory T cells and the interaction with the blood-brain barrier. So actually from both sides. So I study how regulatory T cells affect the blood-brain barrier, but also the opposite way. So how the blood-brain barrier affects regulatory T cells. So shortly, that's what I work with.
Okay, can I ask you what drove you to study Tregs in the first place?
Yes. Well, actually, I already thought about regulatory T cells in my master my master program. So for some kind of course, we had to write like this mini review, this small review, and I chose the topic of regulatory T cells, which was very interesting to me, and I studied them, or I did a literature review in the context of multiple sclerosis. And this was actually also the topic of my current PI, Bieke. And so she contacted me and she filled me in on their research about the interaction of Tregs and the blood-brain barrier. And then, of course, my interest was instantly sparked. I was like, this is so interesting. So I decided to do my thesis internship at her lab, and then
Nice.
I continued my PhD, and now we're, I think, almost 5 years later in the timeline, and I'm still very excited about the Tregs.
So your master's was also in Belgium.
Yes, yes.
And was it like a master's in immunology or like human biology?
So it's a master's in biomedical sciences
Okay.
with like a molecular mechanism background, and then we had some optional courses which I took indeed in immunology.
Okay. Well, great choice in my personal opinion.
Thank you.
Okay. So can I ask you next, why do you personally think that Tregs are the center of this year's Day of Immunology? And do you agree with it? I kind of suspect you do.
Obviously, yes. Obviously, I agree that they definitely deserve the spotlight. And of course, it's probably due to the Nobel Prize of last year being awarded um, to several researchers who discovered regulatory T cells and FOXP3 role in these cells. And with that, with the Nobel Prize being awarded to that topic, of course, it got a lot of attention. Um, and I'm definitely excited about Tregs being put in the spotlight, both with the Day of Immunology, but also with the Nobel Prize, because I feel like researchers or scientists that knew Tregs also now are more aware of the importance of Tregs and what role they can play. But I feel like also with like non-scientists, that also they got to hear about this cell type that's very important in your immune system. And so I think it's very nice that like now there's a more broad understanding of Tregs, and that a lot of people already, if you hear the words regulatory T cell or Treg, that it rings a bell, that at least it got the Nobel
Okay.
Prize, so it must be important, right? Which
Yes.
it definitely is.
So basically, it's a long-awaited spotlight, let's say it like that.
Yeah, yeah.
Uh, so the next question I have for you is, what do you find most exciting about Tregs and compare in comparison to other cells? Because I, for example, I love macrophages. Like, what does make a Treg more interesting than a macrophage?
Well, I don't think I can probably convince you. Um, but I mean, there's a lot of exciting things about Tregs. I think what's the most exciting is like their multifunctionality. Like, of course, they are mainly known as like suppressors of other immune cells, like B cells, APC. But besides that, they have so much more functions within the complete body, like in several parts. Like they induce tissue regeneration, they can help wound healing. And now we actually discovered also in collaboration with a team from Ghent University that they are also important for brain barrier integrity. So in all parts of the body, they play very important roles. So I think that's the most cool thing, the most exciting thing compared to other immune cells. Like other immune cells, they're pro-inflammatory, they help probably with like the virus or bacteria infection, but Tregs are so much more, I think.
Okay, I kind of agree, but I feel like I also feel the same about macrophage. There's so much more. Actually, I have one question about that. If I can skip a little bit, because as I said, I work with macrophages, and people commonly, say they are M1 or M2 subtypes, and I mean, it's very simplified. I do understand it, and it kind of, as you said, it rings a bell already. What kind of cytokines they produce and etc. But do you think with Tregs, people often think that they are just suppressive cells, while in reality they can be much more.
Um, in general, yes. But I think there's more recognition now coming along that Tregs are also more complex than only the immune suppression. And I don't think personally, I mean, I've not been that long in the field, but I never experienced that it was that black and white in the Treg field. I feel like indeed with macrophages it's very one or the other, but I don't think we ever thought about Tregs
In
in
the
that way,
same
like
way.
one or the other. I think it's always been known that it's like quite fluid.
Yeah, so it's like a gray area. And I
Yeah,
mean, with all of the sequencing and new methods coming in, we're just seeing how plastic and different all
yes.
of these cells can be. I agree, I agree. So I have a very interesting question is like, what makes a Treg a Treg, except just the phenotype? If you were like talking to a child, a 5-year-old, and like, what would you say makes the Treg a Treg?
That's a good question. Of course, the first thing I would say is FOXP3. That
Yeah.
makes me like, um, but besides that, if I would explain it to like a child or someone who doesn't know anything about immunology at all, I think I would describe the Treg as like the good guy
Oh,
amongst T cells. Like, I mean, this is the case in my research. I do research in
immune
autoimmunity and
disease, yeah.
neuroimmunology. So in these fields, of course, we think of the Treg as they're the good guy, they will solve everything
Yeah.
compared to all of the other bad immune cells. So it sometimes makes me think about like that one guy in the rom-com movies that's like part of the cool gang but still stands up against the bullies.
Oh my
So
God, I love that. I love that analogy. Because for me personally, because I work with cancer, obviously the first instinct would not be a good guy. So I
no,
find
yeah.
this to be a very, interesting kind of different perception. But I think like if you're talking to a kid, a 5-year-old, seeing a
Mhm.
good guy rather than a bad guy might be a little better.
Yes. Indeed, it's a bit the cancer field and the autoimmunity
Yeah,
field. They have like completely opposite views on Tregs. So
yeah.
it's only from the field that you're looking at.
Maybe they will match sometime in the future. The future Nobel Prize winner might figure out how the theories are basically the same. Probably not, but like we're spitballing here.
Yeah, who knows? Who knows?
Um, okay, so in your specific research, was there any moment when you like stopped yourself and you were like, "Oh my God, this is really cool."
Definitely like multiple moments, but I mean one experiment I can think of is in context of like this paper that we're currently finishing up, which is on the brain barrier protective role of Tregs, which we think goes via IL-34 expression. And like since the beginning of this project, we had the idea of creating these conditional knockout models in which we knock IL-34 out from the regulatory T cells. But I mean, mouse breeding, it took so much time, and there were some issues. So it took 1.5 years before we could actually do the first experiment with these mice.
Oh my God.
And then the first experiment, we looked at just in a steady state whether the brain barriers are more or less leaky if we knock IL-34 out from Tregs. And we actually saw that when in this conditional knockout model, that the brain barriers were so much more leaky compared to their controls. And it was so cool for me, like, it's unbelievable that like one cytokine knocking out from like this small immune cell subset that could have such a big impact. So that was really cool to see it actually having such a big impact, and to understand like, oh, these Tregs are so important in our just normal day-to-day or normal homeostasis of the body.
It's always interesting how like this tiny little thing can have such a broad effect. Yeah, I agree. And you say, I just have to comment on this, is like you had 1.5 years, you said, to do this experiment. And then when people ask, why is your PhD taking so long?
Exactly.
Just stop. Just stop.
Yeah. Exactly.
But it sounds really fantastic. And I mean, after 1.5 years to see like that there is such a difference with IL-35 for knockout, I mean, yay. And I mean, I believe that was quite exciting. And thank you for sharing this with us. Oh, going back to them being, you said they are very flexible, and but sometimes they can also be stable. Sometimes they change, sometimes they regain. Is this what keeps the Treg field exciting for you, or is there something else we don't know about?
Yes, it definitely keeps the field exciting, but
Mm.
also I feel like it keeps it a bit difficult.
Yeah,
It makes
I
it
can imagine.
yes. So definitely researching these cells and doing experiments with them. It makes it sometimes a bit difficult because you want to do some ex vivo experiments. We isolate Tregs from human blood, and then we have to make sure that they stay Treg, of course. So that makes it a bit difficult that they don't already like switch before you add some kind of treatment or do whatever with them. And actually, we are also researching this, how stable they are after migrating through the blood-brain barrier. So we saw that these regulatory T cells, after they migrate through the blood-brain barrier, that they actually have a phenotype switch, so that they become pro-inflammatory, that they lose their suppressive phenotype, they lose some regenerative markers. So they are indeed very flexible, and it's very interesting to see what changes this, of course, how you can skew from one to the other, phenotype, let's say. So yeah, it makes the field interesting, but also complicates things.
Yeah, I feel like that's a common thing with immunology, is like it's a very exciting that things change, but it's also like, can you maybe not sometimes? Following on this as well, because you shared the thing that was very cool was the IL-34 knockout. So was this also surprising for you, or is there something else that you have also experienced with Treg that surprised you in a sense that you did not expect this to happen, and then suddenly they were like, well, look at me, this is what I can do.
Yes, well, indeed, it's also a bit within the same project, but more in the beginning of this project. Um, so maybe like a fun anecdote. So at like this brain barrier symposium that we have here in Belgium, we have like this symposium about all about the blood-brain barrier and the blood-CSF barrier. And we saw some people that looked at the blood-brain barrier um, when all lymphocytes were depleted, so in like these Rag2 knockout mice. Um, and then they also depleted like T cells or B cells because they wanted to know like if we deplete these cells, like do we see something changing in the blood-brain barrier? And they presented this research and they were a bit stuck. They were like, yeah, we see these differences if we deplete T cells, but we're not sure how to continue this project. And then we saw their presentation and we were like, yeah, we probably think it's a Tregs. So we suggested it to them, and then they did like this Treg depletion in mice, and they indeed also saw that the brain barrier was less integral, that it was disrupted. And then that was actually, I mean, it was something that we thought of, but it was very surprising to us that it was actually the Tregs, that they have had such a big effect. So that was what actually was surprising to us. But of course, it's the T-Rex, of course.
Of course. Well, it also shows like how the networking is important and going to conferences and yeah. Oh, this is so
Definitely.
quite surprising and quite nice. Just like depleting the T-Rex can have this effect because you haven't done the full T-Rex depletion in your lab. You were just knocking out specific cytokines like IL-34, right?
Yeah, so this was the T-Rex depletion was actually already a couple of years ago, and then we
Okay,
decided to continue on this using the knockout model.
Oh,
Yeah,
oh,
yeah,
okay, okay,
yeah.
interesting. Um, so okay, so going back to your blood-brain barrier, as you said that like when they cross it, they change the phenotype. So do you think that Tregs are more defined by where they are than what they are? It's a little bit of a strange question, but I'm looking forward to your answer.
Um, yes, it's a very good question. Also, like tissue homing,
Mm.
um, is of T-Rex is definitely a hot topic at the moment, I feel like. Um, so my answer would be like partly yes. Um, so it is known that T-Rex of different organs have like this a bit of a different transcriptome. They change a little bit, but I think of course their core identity and their core function stays stable. I feel like it's a little bit the same with humans, like whether you are located in Africa, America,
Still
Asia, Europe,
human.
like we all look a bit different. We might have a bit of a different culture, but like our core, we're all still human. So I feel like it's the same with T-Rex, whether they are in the brain, whether they're in the liver, in whatever, in the guts. Of course, they change a bit, but they mainly stay the same, I feel like.
In the core, they are the same
Yes.
FOXP3 cell.
Yes.
Good guy.
Yeah.
Yeah, I
Yeah.
really like the human analogy. I think this is really, really clever to show a T-Rex as, living in different tissues slash countries. I might use that sometimes in the future. So as you said that you're working with autoimmune diseases. So one of the questions I have to ask you is because you said they also change, like become more pro-inflammatory. I think in the paper you use like you call them effector then or something
We
like
call them
T.
X-FOXP3 cells.
Oh, okay, okay.
Yeah.
So do you think that we will be able to fix them in autoimmune diseases like multiple sclerosis, or do you think it's a bit more complicated than that?
So I think we can for sure enhance their function.
Mm,
I've seen several labs attempting to like rescue Tregs in these diseases. They have tried different strategies, and I've seen a lot of promising stuff. So I
mm,
definitely think we have some ability to um, get their function back to a sufficient level to use them as a therapy.
mm,
Whether we will be able to fully fix them, I doubt that personally. I think there are too many like unknown variables,
mm,
like environment, genetics. Like for example, one of the research groups here in Hasselt University discovered that like a high salt diet can affect the Tregs. Like a lot of papers are published on the genetic makeup of some people that can change their function. So I think it will be very difficult to figure out what is exactly at the base of these Tregs being dysfunctional. So personally, I don't think we will be able to fix them for like 100%, but I'm
mm.
very positive that we will have some strategies to enhance their function enough to see them, to use them as a therapy, and to have, yeah, a therapy that's can be used very broadly for several diseases. Yeah.
That's very interesting, and I actually have a follow-up question. Is because we see them lose their function, but they can also regain it. Does that give you some kind of hope for therapies in the future?
Yeah, definitely. Indeed, we can tip them one way or the other.
Mm,
I think we just need to find a way to get the balance towards their positive functions.
mm,
Of course, in autoimmunity, that's like their suppressive function. In cancer immunology, it can be the other way around. But I think the more research we do on these Tregs and the more we get to know them, what kind of environment gets them one way or the other, we can of
mm.
course manipulate that and use that to our advantage in therapies. Yeah.
That's very interesting. Can I ask you, like, which this is a little bit maybe off topic, but I find it interesting to see which type of methods do you think that we are currently gaining more and more, like single cell sequencing or spatial sequencing? And there was even, I think Singleron had these like single cell sequencing, but also coupled with TCR, for example, for the T cells, like they can have the protein as in the same scenario. I don't really know specifically how the methods work, but like in the field of Tregs, which kind of like avant-garde method do you think will be most used or most beneficial in the future of research? Maybe for younglings that are looking into doing a PhD on Tregs, like what should they know?
Yeah. That's a good question. I mean, all of the methods that you describe are, I think, very useful. I think indeed the TCR sequencing is something that we should keep an eye on, especially in like the field of autoimmunity, um, to get like to know um, how we can get these autoreactive Tregs
Mm,
to like the autoantigen, um, what they do and how we can use them to our advantage
mm,
um, in the field of like therapies. But of
mm.
course, a combination of all
A
the,
combination is always a good thing.
yeah,
I
of all
mean,
the
like I
aspects
feel
indeed.
like in recent times I have seen more and more statements like immune informatics, not just bioinformatics, but we are going into immune informatics as well now. So I feel like whoever is going into PhD or any kind of research at this stage, they should have a little bit of insight into any kind of sequencing because it's getting so big, honestly, the whole
Yeah,
field. Yeah.
definitely, yeah. Sometimes I get a bit lost in it. I'm not
Yeah,
really
I
home into
agree.
the bioinformatics stuff, and I try to understand it all, but sometimes I like it. This is, yeah, it's very cool. You get a lot of information.
Yeah,
So it's definitely, yeah, it's definitely like I think the field will go
even
straight
further.
ahead with these
Yeah.
techniques, which is very nice. But yeah, indeed, people that step into a PhD, it might be nice to have like a bit more background on these techniques. Yeah.
Oh, thank you for sharing that. So in your paper, you were also showing that Tregs can support the brain barrier integrity. So, going into this, did you expect them to have this kind of a non-immune like tissue repair function, or was that something that was just like, oh, look, they can also do this as well?
Yeah. So I mean, we were not very surprised, right?
Okay.
It's already known that they have like these indeed tissue regenerative functions. Um, so when our collaborators on this project presented that they saw like these brain barriers that were leaky in RAC2 deficient knockout mice, and if they did a T cell depletion, these barriers were leaky. During that presentation, I saw my PI and my postdoc who both work on Tregs. I saw them make eye contact, and I for sure know that they both thought like this must be Tregs. It cannot be
Yeah.
another way. So after that, we started like this collaboration, and then yeah,
Mhm.
now the project is almost rounded up. But yeah, I mean,
Congratulations.
thanks. But we were not that surprised. We kind
Because
of
you were like, if they can do it elsewhere, probably in the blood-brain barrier, they can do it as
yeah,
well. Yeah.
yes,
So it was more of an expectation rather than a surprise.
yes, yes,
Okay.
indeed.
Okay. Well, good that they provided, the theories provided
Yeah.
what they were expected. Um, I have two last questions for you that will be a little bit not necessarily very on topic. That one of them I really need to ask. Like we were talking about what was interesting, what was exciting, what drove you to study them, but like what is the frustrating thing about working with T-Rex?
Oh, good question.
You
So,
mentioned before that they are difficult sometimes, so obviously there is something
yeah,
there.
so they can definitely be sometimes a bit frustrating to work with. I mean, I love them, but
Okay, it's understandable.
yeah, yeah.
Yeah.
So I think the most frustrating thing is definitely like the low number, and
Mm,
the mostly the fluctuating cell numbers that we get
mm,
each time. Um, so yeah, we almost always do this like standard T cell receptor stimulation using like
mhm.
anti-CD3 and CD28. And in the lab, we coat the plates for anti-CD3, and they have to be coated like a couple of hours. And I feel like the T-Rex feel if I coat a lot of wells, I don't have, I almost have no T-Rex, so I wasted a lot of the coating. And when I coat only a couple of wells, I have way too many T-Rex. So
Yeah.
it's definitely frustrating that it's like
Yeah.
fluctuating. Yeah, we use a lot of human blood, so it's
Hmm.
depending on the donor, depending on the day. I don't know what it is,
Yeah.
but yeah, that I think that's definitely the most frustrating thing. After a long day in a lab, and then you sorted them, and you only have like 200,000 T-Rex, and I'm like, I cannot
Oh.
even do one experiment with these. So yeah.
I'm the kind of a person who is, if we have two benches or like two hoods, I have to do the whole experiment in one. Otherwise, something will go wrong.
Yeah, me
Talking about
too.
superstition inside.
Yeah. If an experiment one time works out, I'm like, I have to do it exactly like this
Everything.
again.
Just keep it the same.
I
I
completely
agree. I
understand.
agree. Can I ask you also one other off-topic sort of question? Because you said you work with human blood, and I've also seen you work with mouse models as well, which obviously with the immune system it's very relevant and needed. I don't know, have you seen, but end of last year, I think like December, FDA announced like a switch toward more like human relevant models like organoids and spheroids and stuff. That I personally work with organoids and microfluidic devices, so it was very exciting thing for me. Yeah, but how do you think it will affect the field of, let's say, Treg specifically, but in generally immune, because its immune system is like the whole body. Like, do you think that we will be able to do some studies? Like, I was able to look at the tumor-associated macrophages specifically in these systems, but like anything that comes, homing or it might be a bit difficult. So what is your opinion in that, if I can
Mm.
ask you?
Um, I mean, I feel like a lot can indeed be transferred from like animal work to
Mm,
like in vitro work. Um, we in our group we also try to make like indeed we do some mouse work, but then we always try to validate in human models. So for Tregs, indeed we isolate regulatory T cells from human blood, and we do some experiments with that.
mhm.
But I feel like you can, with the immune system, indeed, you can only go that far. So I feel like maybe like 80% you
Mm,
can do like in vitro, ex vivo. Um, but the other 20%, I'm really not sure if we can ever fully translate that to like
yeah.
a dish model, let's say, because indeed it's so complicated. Like we also talked about these Tregs being so flexible.
Mm,
That also goes for like, yeah, indeed tissue homing. So if they arrive into a tissue, they change their phenotype and their function. If they come into contact with other immune cells. So I feel like we will, of course, we can reduce the amount of animals that we use, but I don't think we're that
fully.
far yet to
Mm.
fully exclude them. Yeah.
No, I agree. With immune system, it's particularly difficult because, especially because you know where the cells are coming from. There have been companies that have generated these like multi-organs on
Mm.
chip systems, which can help in
Yeah.
some
Yeah.
way. But the future will tell. Obviously, I personally would, and I believe you as well, would love to see it 100%.
Of
I
course, of
don't think
course.
it will happen that soon, but maybe in the future we get to be hopeful.
Yeah.
Um, I have one last question for you, and it's a bit of a strange one, but I'm a strange person, so I think it's all right.
Okay.
I really like the movie Inside Out. Have you seen that movie?
No, I haven't.
Oh, you have not. Oh my God.
But
Okay.
I
Well,
know the concept.
I will still ask you the
Yeah.
question, but it's a Pixar movie about the emotions in one child's head, and how, she's going through the different years of growing up, and then you have
Mhm.
joy, you have sadness, you have anxiety. Anxiety was Ginger, and I feel personally attacked because I'm slightly Ginger. But as a PhD student, I also correlated with that. But so if you had to pick an emotion for Treg, which one would you pick?
It's it's a very difficult question. I feel like, um, I mean, maybe, yeah, I like T-Rex, so I could believe they could be happy.
Oh,
Yeah, but I mean, sometimes I do compare them. It's not an emotion, but sometimes I compare them to like this toxic ex. They can, whether sometimes they can make you so angry when you've been in the lab for a long day, and
mm.
then you still have too little cells for your experiment, but then you can't stay away from them because you love them so much, and yeah,
So they bring you joy.
inside
They are
there's
joy.
yeah, yes. So inside they're so good. Yeah.
I
So
love it. I love that they are joy. I think for me also macrophages would be joy, a
yeah.
big Pac-Man joyful cell,
Yeah, actually, yes.
right?
Yeah. Yeah, I can see that.
Okay, well, I would recommend the movie. I don't know if you like animated movies, but that one was really nice for me.
Okay, I
So,
will look it up.
yeah, if there is one thing you get from this interview, it's like that one. I'm just kidding. But this was actually my final question for today, and I
Okay.
had so much fun talking to you today. I
Me too,
learned
Nani.
so much because I am not a Treg expert. For me, they are, just one of the. No, they are not. I'm just kidding. But it was very, very nice, very insightful talking to you today, and thank you so, so much for making this day of immunology better for us all.
Thank you for inviting me and letting me speak about my favorite cell type, the Tregs.
Okay, let me just. I think I stopped the transcription. No, I did not.